What are a few of the big challenges materials science researchers are working on now?
One challenge is to make materials that will allow us to start making smaller, lighter cars that can more easily be powered by electricity. LED lights are another big area in materials science right now. We need to develop materials that can use energy efficient LEDs to produce the kinds of light we need at low costs. One last area to mention is reducing the amount of waste we produce in general. There are many approaches to reducing waste. And from a materials science perspective, we would like to make products out of long-lasting, durable materials that can be repurposed or reused at the end of a product's lifetime.
What makes a bowl microwave safe?
Unfortunately, the only definition is an empirical one. Containers that don't get hot in the microwave are microwave safe. Remember, or go look it up now in the world around us, that microwaves heat food by causing molecules with dipole moments to tumble back and forth. If the container has any such molecules, it'll get hot in the microwave. If any water has leached into your ceramic mug, it'll get hot in the microwave, regardless of whether the container gets hot on its own. The food you are heating up will get hot and transfer that heat to the bowl. So be careful when taking hot things out of the microwave.
What is a vacuum?
A vacuum is space without matter. The word vacuum derives from a Latin word that means empty. A perfect vacuum, or one with absolutely no matter in a given space, is very hard to achieve, but through modern engineering scientists can get pretty close without having to go into deep space to run their experiments.
Do you notice a change in the part that you dipped into the solution?
Place the remaining pennies into the solution. You will likely notice a visible reaction as the pennies are placed in the solution. The reason pennies eventually begin to appear dull is that the copper surface reacts with oxygen in the air to create a layer of copper oxide. In this experiment, the vinegar and salt will react with the copper oxide and remove it, which will leave a layer of the original shiny copper exposed on the surface. Allow the pennies to remain in the solution for several minutes. If necessary, try to move the coins around so as to expose both sides of each coin to the solution. If possible, flip the coins over after a couple of minutes. Drain the solution and rinse the coins with clean water. They should now look clean and shiny.
Why did Ahmed Swail, Egypt/United States, win the Nobel Prize for Chemistry.
For his studies of the transition states of chemical reactions using femtosecond spectroscopy. Zuhail was awarded the prize for his work using ultrafast femtosecond spectroscopy to study chemical reactions in real time. That is to say, on the timescale on which they actually take place. Zuhail's earliest experiments in this area looked at iodocyanide and sodium iodide. He was the first to be able to observe the dissociation and recombination of a chemical species as it actually took place. His great advances in spectroscopic techniques revolutionized the field, and continue to do so today.
What determines whether a cell is a skin cell, blood cell, etc.?
The process that new cells undergo to become a specific type of cell is called cellular differentiation. Interestingly, different types of cells don't contain different genetic information, but rather they just express different parts of their genetic information. Signal molecules tell the cell which parts of its DNA to express, which in turn controls what types of proteins and other molecules are present in the cell, and ultimately these factors determine its function.
What molecular properties lead to lower melting points?
Most of the trends we talked about for raising boiling points hold true for melting points for most of the same reasons. There is one major exception, though. The more branched, and therefore compact, a molecule is, the higher its melting point. Because, in general, compact molecules will pack better in a crystal lattice. The better packed a lattice is, the more stable it is, and the more heat energy it takes to break up that lattice and melt the solid.
What is an elimination reaction?
A reaction in which two ligands or substituents are removed from a molecule.
What is chirality?
Chiral objects have non-superimposable mirror images.
How are elements named?
The names of elements often have interesting origins. They have been named after people, places, colors, mythological creatures, or for a variety of other reasons. Some are named after scientists, such as curium, after Marie and Pierre Curie. Lawrencium, Ernest Lawrence, Seaborgium, Glenn Seaborg, Mendelevium. Dmitry Mendeleev, Einsteinium, Albert Einstein, and barium, Niels Bohr. Others are named after places, such as Lutetium. Lutetia means Paris in Greek, Californium. Berkelium, Berkeley, California, Americium, Dubnium, Dubna, Russia, Hassium, Hessen, Germany. Yttrium, Ytterbium, Terbium, and Erbium, these last four being named after Ytterby, Sweden. Tantalum, Tantalus, Niobium, Niobe, Promethium, Prometheus, Uranium, Uranus, Neptunium, Neptune. Plutonium, Pluto, Palladium, Pallas, and Cerium, Ceres, are all named after mythological creatures. Though elements can take on different names in different countries, the commonly accepted names are those agreed upon and assigned by the International Union of Pure and Applied Chemistry, IUPAC.
What is a carbon-hydrogen bond activation reaction?
Carbon-hydrogen activation reactions are very much what the name would imply. They break typically unreactive carbon-hydrogen bonds. Considering the prevalence of carbon-hydrogen bonds in organic compounds, this is another extremely important type of reaction to be able to carry out both effectively and with selectivity. Successful examples of carbon-hydrogen bond activation reactions have only come about relatively recently, with the first genuine example reported ca. 1965, and organometallic reagents have played a key role in the development of carbon-hydrogen bond reactions.
Why did Alan J. Heeger, United States, Alan G. McDermott, United States/New Zealand, and Hideki Shirakawa, Japan, win the Nobel Prize for Chemistry?
For their discovery and development of conductive polymers. This prize was awarded for the discovery of plastics/polymers that can, under certain circumstances, be made conductive. This was quite a surprising result, since nobody ever expected plastics to be conductive materials. These plastics/polymers consist of chains of conjugated carbon-carbon double bonds. In other words, carbon-carbon double bonds that alternate with carbon-carbon single bonds. These polymers were also doped, which means that electrons were either removed or added artificially by oxidation or reduction.
How is refined sugar different from raw sugar?
Refined sugar is raw sugar that has been purified by a series of steps that ends with crystallization of a sugar syrup into white sugar crystals. The debate of whether refined sugar is worse for you than raw sugar is ongoing. But chemically refined sugar is just more pure sugar than raw sugar.
How are formal charges different?
Formal charges are given for individual atoms within molecules. These are determined by dividing the electrons in every bond equally between the atoms that share them, regardless of the elements involved.
What is an electrolyte?
An electrolyte is a substance that contains ions, charged particles, and is capable of conducting electricity. Most often this is a solution containing dissolved ions, like an aqueous solution of sodium chloride, which contains Na and N-clions. The presence of ions in the solution is what makes it capable of conducting electricity. It's for this reason that it can be dangerous to get the electrical outlet wet when drying your hair in the bathroom. Pure water itself doesn't conduct electricity, but in practice, there are almost always some ions present, making tap water an electrolyte. Another place electrolytes are important is in living things. Fluids in the human body, and in other organisms, contain free ions and are thus electrolytes. Gradients in ion concentrations in cellular fluids are often important for regulating cellular processes and bodily functions such as muscle and nerve activity.
What is an atomic orbital?
An equation that describes the probability of finding an electron around a nucleus.
What is a colloid?
Suspension of particles of one substance in another, e.g., milk.
What do the different octane ratings mean at the gas pump?
The octane number provides a measure of the amount of compression a fuel can undergo before it combusts. The name octane rating comes from the fact that isooctane is used as the standard against which a fuel's compression characteristics are measured. A fuel with an octane rating of 89 means that it has the same ability to withstand compression as a mixture of 89% isooctane and 11% heptane. In reality, gasoline contains many other components. Fuels with higher octane ratings are typically used in higher performance automobiles. Since fuels may be able to withstand compression better than isooctane, it's entirely possible to achieve octane ratings of over 100.
What was the law of triads?
A scientist named Johann Dobereiner discovered trends in the reactivity of groups of elements. Certain sets of 3 elements, such as lithium, sodium, and potassium, showed similar chemical properties. And Dobereiner noticed that the average of the atomic masses of the atoms of the heaviest and lightest elements in the triad gave the atomic mass of the midweight element. For example, the mean of the atomic masses of lithium and potassium is 3 plus 19/2 equals 11, which is the atomic mass of sodium. Due to differing numbers of neutrons in each element and the existence of different isotopes. This law isn't always strictly true, but it does tend to work well, especially for lighter elements. For reasons we hope to explain in the coming questions. This trend plays an important role in the structure of the periodic table.
Why did Ernst Otto Fischer, Federal Republic of Germany, and Geoffrey Wilkinson, United Kingdom, win the Nobel Prize for Chemistry?
For their pioneering work, performed independently on the chemistry of the organometallic, so-called sandwich compounds. Fischer and Wilkinson received the prize for their role in elucidating the basic properties surrounding bonding and reactivity in organometallic complexes, with a particular focus on their work with organometallic sandwich compounds, which involve a metal center sandwiched between 2 ligands. This prize was somewhat special in that it was openly recognized that the practical implications of their work were not readily obvious, but that the knowledge surrounding organometallic chemistry gained from their work would be invaluable to chemists in the future. Why did Paul J.
What is echinacea?
Echinacea is a group of flowers, related to daisies, sometimes also called coneflowers. Different members of this group are used in herbal medicines to stimulate the body's immune system. Scientific studies on the effectiveness of echinacea are contradictory, however, as some have shown clear effects in the prevention of or shortening of the length of colds, while others have concluded that it is mostly ineffective.
What is a quark?
Quarks are the fundamental particles that make up protons and neutrons, as well as several other types of particles. There are 6 types of quarks, which are referred to as different flavors. These are named up, down, top, bottom, charm, and strange. Protons and neutrons are each made up of 3 quarks. 2 up and 1 down quark make up a proton. 2 down and 1 up quark make up a neutron.
Why did Karl Ziegler, Federal Republic of Germany, and Giulio Natta, Italy, win the Nobel Prize for Chemistry?
For their discoveries in the field of the chemistry and technology of high polymers. These two great polymer scientists developed several classes of polymers, while simplifying and clarifying the mechanisms of polymerization processes. Ziegler discovered titanium complexes that can catalyze olefin polymerization reactions. And Natta developed a method for the preparation of stereoregular polymers from propylene. At the time, the Nobel Committee recognized that the full implications of their work were likely not yet realized. And indeed, polymer chemistry was still a relatively young field at that date. Today the work of Ziegler and Natta underpins the technology used to generate many of the plastics you encounter.
Is there water on other planets?
There is, though in most cases where water exists on other planets it does not exist predominantly in the liquid form, like here on Earth. On some planets there may be trace amounts of water vapor in the atmosphere. Beds of ice on a planet's surface, or superheated, ionized water near a planet's core. There may well be other planets out there with liquid water. But humans have not found many with large amounts of liquid water.
Why did Carrie B. Mullis, United States, win the Nobel Prize for Chemistry?
For contributions to the developments of methods within DNA-based chemistry for his invention of the polymerase chain reaction. PCR, method, and Michael Smith, Canada. For contributions to the developments of methods within DNA-based chemistry for his fundamental contributions to the establishment of oligonucleotide-based site-directed mutagenesis and its development for protein studies. Mullis was awarded the prize for developing a widely used laboratory method called the polymerase chain reaction, PCR, method. PCR allows the use of relatively simple lab equipment to generate millions of copies of a sample of DNA in the time span of only a few hours. This has found diverse applications in a large number of laboratories. It can be used, for example, to generate copies of DNA from fossils of organisms that are long since extinct. Smith shared the prize with Mullis for his work manipulating the genetic code. In cells, proteins are produced based on sequences of nucleotides contained within the organism's DNA. These sequences dictate the sequences of amino acids that will be incorporated into a protein. Smith developed a method to selectively manipulate DNA to alter the amino acid sequences of the proteins that are produced.
Brown, United States, and George Wittig, Federal Republic of Germany, win the Nobel Prize for Chemistry?
For their development of the use of boron and phosphorus-containing compounds, respectively, into important reagents in organic synthesis. Brown received the prize for his work with boron reagents in organic synthesis, which also led to the development of organoborans as a class of molecules. Wittig worked with phosphorus, developing a reaction through which a carbonyl could be converted to an olefin. This reaction is today known as the Wittig reaction. The work of both Wittig and Brown resulted in the development of useful reagents that find wide application in organic synthesis even today.
What is a racemic mixture?
A racemic mixture contains equal amounts of both enantiomers of a molecule.
How do mood rings work?
Mood rings change color based on your body temperature. The piece that changes color is a liquid crystal thermometer, which is the same technology that is used in some disposable medical thermometers as well as the adhesive thermometers commonly used in aquariums.
Is it true fresh eggs will sink in water, while bad ones will float? Why?
Yes. As an egg goes bad, proteins and other chemicals decompose and release volatile molecules like carbon dioxide, CO2. The shell of an egg is somewhat porous, so these gases can escape, decreasing the mass of the egg. Since the size of the egg doesn't change, assuming you don't break it, the density decreases as the egg ages, and it eventually becomes less dense than water. Whether it becomes buoyant exactly when it is spoiled or weeks after, we can't say. That's why your nose is so good at smelling that awful smell so that you don't eat rotten eggs.
What is flame ionization detection?
Flame ionization detection, FID, is a method of detecting organic analytes in gas phase chromatography experiments. A very hot flame is used to burn the analytes emerging from the chromatography instrument, which produces positively charged ions. These ions are then attracted to a negatively charged electrode, which detects them by generating an amount of current proportional to the number of positively charged carbon atoms reaching the electrode. FID can be particularly useful due to the fact that it detects organic analytes without any interference from a wide variety of other gases, which can be present in the sample or in the carrier gas used in the gas chromatograph. There are some downsides to FID, one of which is that the sample is destroyed as it is analyzed.
What is plasma?
Plasma, the fourth state of matter, is a gas where some amount of the particles have been ionized. As little as 1% ionization leads to very different properties for plasmas than gases, including increased conductivity, like lightning, and magnetization.
How fast is Earth moving through space?
When taking into account the motion of the Milky Way galaxy, the fact that our solar system rotates within this galaxy, and the motion of the Earth within our solar system, it is estimated that the Earth is moving at about 500 kilometers per second. Of course, since everything around us is moving at the same rate, we don't tend to notice this.
What is a halogen?
Elements in group 18, group VIIA.
So is there a tannin taste bud?
No one knows yet.
Why do some chemical reactions cause a color change?
The colors we see all have to do with what wavelengths of light something absorbs or reflects. For a chemical reaction to cause a change in the color of something, all that has to happen is that the products of the reaction absorb and reflect different wavelengths of light than the reactants do. We'll discuss how light interacts with molecules in more detail in physical and theoretical chemistry.
What is a van der Waals force?
Intermolecular forces between molecules resulting from the interactions of dipoles or induced dipoles.
What is an antiparticle, and what is antimatter?
For most kinds of particles there is postulated to exist a corresponding antiparticle, which is of the same mass but an opposite charge. These antiparticles have only recently been observed in laboratory settings for the first time, and they are very difficult to isolate and study experimentally. This is because particle and antiparticle pairs collide to generate photons of light in a process that annihilates the particle and antiparticle pair. Antiparticles are not well understood and are an active area of research related to nuclear chemistry. Antimatter is just matter made up of antiparticles. in the same way that normal matter is made up of particles. There has been postulated to be an equal amount of matter and antimatter in the universe. Though the observations made to date do not suggest this to be the case. This represents an unresolved dilemma that scientists hope to someday better understand. These types of fundamental, unresolved problems are a big part of the reason science is so interesting.
What is a chirality?
A geometric property of a molecule whose mirror image is non-superimposable with the original molecule.
What is the condensation reaction?
A reaction in which two molecules combine to form one larger molecule, with the concurrent loss of another small molecule like water or HCl.
What is blackbody radiation?
Electromagnetic radiation given off by a black body. At room temperature, most of this radiation is in the infrared. But at higher temperatures, visible light can be emitted.
Does hot water really freeze faster than cold water?
Sometimes. This observation is known as the Mpemba effect, named after the Tanzanian student who in 1963 resurrected the idea from Aristotle, Francis Bacon, and René Descartes. Whether or not this effect can be seen seems to depend on so many variables: the size and shape of the container, the initial temperatures of the two liquids, the method of cooling, on how you define freezing, when the first ice crystal forms, when there's a solid layer on the top, or when all of the water has frozen solid, that it's really still unclear if this effect is real or not.
Can I think of chemical bonds as springs between atoms?
Chemical bonds can be thought of as springs holding together the atoms in a bond. When atoms in a bond are stretched or compressed from their equilibrium separation, the bond provides a force to pull the atoms back together or to keep them from getting too close. For relatively small displacements, the bond actually provides a force that is physically very similar to that of a spring connecting two objects. This model of a spring as a chemical bond can be very useful for getting an intuitive idea of how a chemical bond connects atoms in a molecule.
What does brining do to meat?
Soaking meat in a salt solution, the definition of brining. Helps separate the long filaments that make up muscles, myofibril, by dissolving the proteins on their surfaces. With enough salt the actual filaments start to break down, and both effects make the meat seem more tender. Additionally, salt allows proteins to hold onto more water, which helps prevent your steak from drying out.
Why did Victor Grignard, France, win the Nobel Prize for Chemistry?
For the discovery of the Grignard reagent and Paul Sabatier, France. For his method of hydrogenating organic compounds in the presence of finely disintegrated metals. Grignard was recognized for his preparation and study of organic magnesium compounds. RMGX, which he prepared from organic halides, RX, and magnesium metal. He discovered that these magnesium reagents can react with carbonyl groups to form new carbon-carbon bonds. Sabatier shared the prize in 1912 with Grignard for his work on hydrogenation, or the addition of a molecule of H2 to an organic molecule. Sabatier observed that nickel acted as a catalyst for these reactions. The Sabatier process, the reaction of Co2 and H2 to form CH4 and H2O, continues to be a useful reaction. And NASA is investigating it as a means to generate water from CO2 waste, from exhaling astronauts.
What is a solvent?
The major component of a solution.
How does photosynthesis work?
Photosynthesis is the process carried out by plants to harvest energy from sunlight. The most important molecule in photosynthesis is called chlorophyll. It's what collects sunlight and gives plants their green color. Carbon dioxide, CO2, is taken in through cells called stomata. And the plants also draw water up through the roots and into the leaves. The reaction catalyzes when chlorophyll absorbs light and makes ATP along with another molecular energy source called NADPH, nicotine adenine dinucleotide phosphate. In the process, CO2 is used up and water molecules are split, releasing O2 gas, which other organisms, like humans and animals, can then breathe.
How much energy in the United States comes from hydroelectric power sources?
About 10% of the electricity in the United States comes from hydroelectric power sources. The state of Washington leads the United States in hydroelectric energy production, with roughly 87% of the state's electricity coming from hydroelectric power.
What is a positron?
A positron is the antimatter counterpart of the electron. It has the same mass and spin as an electron, but with a charge opposite in sign and equal in magnitude to that of the electron. If an electron and positron collide, they can annihilate each other and release their energy in the form of a photon.
How dense is mercury compared to other liquids?
The density of liquid mercury, it is a liquid at room temperature, is about 5.43 g/ml. This is about 5.5 times as heavy as water. One of the next most dense liquids is bromine with a density of 3.03 g/ml. But mercury remains almost twice as dense.
What is a cation?
A positively charged ion.
What is chromatography?
Chromatography describes a series of techniques used for separating the components of a mixture of chemicals. The most common type of chromatography is column chromatography. This involves dissolving the mixture to be separated into a solution and flowing it over a column of stationary material that interacts to a different extent with each component of the mixture. As the liquid flows over the stationary phase, the components that interact most strongly will be held behind longer, while those that interact weakly will be eluded. From the column first, the liquid is collected into a series of small vials or test tubes, which will, hopefully, each only contain one component of the mixture. The solvent can then be evaporated from each vial to recover the individual components of the mixture. A wide variety of chromatographic techniques exist, and each generally follows the same basic principles as the column chromatography described here.
Why did Theodore William Richards, United States, win the Nobel Prize for Chemistry?
For his accurate determinations of the atomic weight of a large number of chemical elements. Richards was the first American to receive the Nobel Prize in Chemistry. He and his students accurately measured the atomic weight of 55 different elements and also showed that some crystalline solids can contain gases or other solutes within their lattices, technical term, a fluid.
Why did Gerhard Ertl, Germany, win the Nobel Prize for Chemistry?
For his studies of chemical processes on solid surfaces. Surface chemistry is a topic of broad importance to many applied areas of chemistry. These include atmospheric chemistry, as some atmospherically relevant reactions take place at the surface of ice crystals. Semiconductors and solar energy capture, to name a few. Ertl was one of the founding fathers of modern surface science, and he played a role in developing many of the techniques used to study surfaces today. The study of well-characterized surfaces typically requires studies to be carried out under ultra-high vacuum conditions so as to avoid contamination of the surface. To be studied by any gas phase molecules that may tend to adsorb onto the surface.
What is diamagnetic?
A material that creates an opposing electric field in response to an applied electric field.
What is the Maillard reaction?
The Maillard reaction is technically non-enzymatic browning, which basically includes any kind of browning that happens when you're cooking, but excludes what happens to cut apples you leave out on the counter. At the chemical level, it's a reaction between an amino acid and a sugar in the presence of heat. A huge number of chemicals are formed in these processes, so you can't really pin down the Maillard reaction to a single set of chemical steps. These processes are responsible for the browning of meat, the malting of barley for beer, the roasting of coffee, and the browning of the crust of bread.
What is an alkali metal?
Group 1 of the periodic table, i.e., Li, Na, K, Rb, Cs, Fr.
What is para?
Term used to describe two substituents on an aromatic ring that are bonded to positions opposite to each other.
How many nuclear power plants are there in the United States?
There are currently 65 nuclear power plants in the U.S. and a total of 104 nuclear reactors. This is because 36 plants have more than one reactor.
What is a phase boundary?
The interface between two phases, e.g., solid-liquid, liquid-gas, or solid-gas boundary.
What is a racemic?
A mixture of equal amounts of the two enantiomers of a chiral molecule.
Why do mass spectrometers need a vacuum to operate?
As the ions travel, they must not collide with any other atoms or molecules before they reach the detector. If they collide with anything else, this will change their direction and kinetic energy, which will make detection either unreliable or impossible.
What is biocatalysis?
Biocatalysis, as the name suggests, involves using enzymes or other natural catalysts to carry out chemical reactions. This tends to work well within the context of green chemistry, since biological reactions are often catalyzed in water at mild temperatures and pH values. Moreover, enzymes are themselves environmentally benign and obtained from natural sources. In addition to these benefits, biocatalyzed reactions typically proceed with high selectivity and specificity, and require relatively few synthetic steps, thus minimizing the amount of unwanted byproducts produced. Some examples of biocatalyzed syntheses are those of penicillins, cephalosporins. Another class of antibiotics, and pregabalin, a drug to relieve pain from damaged nerves. The use of biocatalysis is gaining popularity as the necessary technology becomes more readily available and as more people realize the benefits of this approach.
How much CO2 production comes from automobiles?
According to the Environmental Protection Agency, EPA, the transportation industry as a whole accounts for 33% of all CO2 emissions in the U.S. Automobiles used for personal transportation account for 60% of this, or 20% of total U.S. CO2 emissions. The remainder comes from other sources of transportation, such as large diesel vehicles and airplanes burning jet fuel.
How can I carry out a chromatography experiment at home?
Chemistry principles encountered in this experiment: extraction, filtration, chromatography. Materials you'll need: rubbing alcohol, 250 ml, small jars with lids, baby food jars work well. Tree or plant leaves, 5 will do. Coffee filters, pen containing hot water, kitchen utensils. The procedure: tear a few leaves up into small pieces, and place the pieces from each leaf into their own small jar. Add a small amount of rubbing alcohol to each, such that the leaves are just covered by the rubbing alcohol. Place the lids on the jars, and then place them in the hot water for 30 minutes. Replacing the water with fresh hot water if it cools. Swirl the alcohol in the jars every 5 to 10 minutes. By the end of 30 minutes, the pigments from the leaves will dissolve, and the alcohol solutions should appear colored. The alcohol serves as a solvent for extracting the pigments in the leaves. Cut long strips of coffee filter paper, remove the tops from the jars, and place one into each jar, with one end in the alcohol, and the other end outside the jar. The pigments will move up the paper different amounts, according to their size. You should be able to see the separation as different colored regions on the paper. Note that this process is a bit different from some of the separations we described in the modern chemical lab based on polarity or other chemical properties. Unlike chromatography on silica gel, the paper is not strongly polar and thus does not interact as strongly with the polar pigment molecules. Remove the strips of paper and allow them to dry. By comparing the relative distances the compounds traveled, technical term, retention factor, of the different compounds with one another, you should be able to identify whether the different leaves contain the same pigments. You can also try to make your alcohol solutions from other pigments, such as those in inks, foods, or drink. Try to design a hypothesis you can test using this experiment, such as whether or not the leaves of different plants contain the same pigments, or whether different markers or pens contain the same inks.
What is metallurgy?
Metallurgy is the branch of science that deals with the properties of metals composed of both pure elements and those of mixtures of metallic elements, which are called alloys. It represents one of the first efforts to manipulate and understand how elemental composition of a substance affects its physical properties.
Why did Carrie B. Mullis, United States, win the Nobel Prize for Chemistry?
For contributions to the developments of methods within DNA-based chemistry for his invention of the polymerase chain reaction. PCR, method, and Michael Smith, Canada. For contributions to the developments of methods within DNA-based chemistry for his fundamental contributions to the establishment of oligonucleotide-based site-directed mutagenesis and its development for protein studies. Mullis was awarded the prize for developing a widely used laboratory method called the polymerase chain reaction, PCR, method. PCR allows the use of relatively simple lab equipment to generate millions of copies of a sample of DNA in the time span of only a few hours. This has found diverse applications in a large number of laboratories. It can be used, for example, to generate copies of DNA from fossils of organisms that are long since extinct. Smith shared the prize with Mullis for his work manipulating the genetic code in cells, Proteins are produced based on sequences of nucleotides contained within the organism's DNA. These sequences dictate the sequences of amino acids that will be incorporated into a protein. Smith developed a method to selectively manipulate DNA to alter the amino acid sequences of the proteins that are produced.
What are some applications of functionally graded materials?
Recall from above that a functionally graded material is one that varies in one or more properties throughout its dimensions. These constitute a relatively young class of materials with promising applications in a variety of areas. For example, the living tissues in your body, including your bones, are classified as natural, functionally graded materials, so if scientists want to develop materials capable of replacing these, they are looking to develop a functionally graded material. They are also useful in aerospace applications, where materials that can withstand a large thermal, temperature, gradient are needed. Functionally graded materials are commonly found in energy conversion devices and have also been used in gas turbine engines. They can also be good at preventing the propagation of cracks through the volume of a material, which makes them promising candidates for defense applications like developing bullet-resistant materials to create armors for humans and vehicles.
What is smoke?
Smoke is a cloud of particles given off by a material that undergoes combustion. Its chemical composition will vary depending on what material is being burned. Smoke may consist of hydrocarbons, haloalkanes, hydrogen fluoride, hydrogen chloride, and a variety of sulfur-containing compounds, among others. These compounds can vary widely in their toxicity, so the severity of the health hazards associated with the smoke from a fire will depend on what is being burned.
What are interferences?
Interferences are substances that interfere with the measurement of the analyte we are interested in. They give a similar response to our analyte in whatever analytical measurement we are making. Typically, analytical chemists want to remove any interferences from a sample prior to making a measurement, or, alternatively, choose the method of analysis carefully, so as to avoid any signals caused by interferences.
What are neurons?
Neurons are cells responsible for transmitting information between different locations in the body. Their functions are diverse and include telling muscles to move, passing information from your sensory organs to your brain, making you experience pleasure, and processing information in your brain.
How does a battery work?
Batteries operate based on chemical redox reactions that are set up to cause the spontaneous flow of electrons through the object you want to power.
How is ethanol produced for use as a fuel?
Ethanol is produced by fermenting sugars from plants like corn, soybeans, and sugarcane. The sugars in the plant material are first broken down and then fed to yeast, which ferments them to produce ethanol as a byproduct.
Can wearing a copper bracelet help with the symptoms of arthritis?
Arthritis is caused by deterioration of the cartilage in a person's joints, where deterioration happens faster than the body is able to repair it. Copper bracelets are sold based on the idea that a person may have a copper deficiency that leads to the pain in their joints, thus implying that copper from the bracelet could be absorbed through the skin to help correct the deficiency. In fact, copper deficiencies are extremely rare, and most people eat plenty of copper in their regular diet. Rarely are additional copper supplements necessary. It has not been proven that copper can be absorbed through the skin, nor has it been proven that the bracelets can help with any symptoms of arthritis or joint pain. Moreover, excess intake of copper can result in poisoning. So if copper can be absorbed through the skin from a bracelet, one would want to monitor their dosage carefully. It is also worth pointing out that we know of no cases of poisoning resulting from wearing copper bracelets.
Why did James Batchelor Sumner, United States, win the Nobel Prize for Chemistry?
For his discovery that enzymes can be crystallized, and John Howard Northrop, United States. And Wendell Meredith Stanley, United States, for their preparation of enzymes and virus proteins in a pure form. Sumner was the first to provide concrete evidence that enzymes and proteins could be crystallized, paving the way for further research in this area. Northrop and his co-workers explored the conditions that led to formation of crystalline proteins, and mastered this art, pioneering the way for modern scientists working to crystallize proteins and viruses. Stanley demonstrated that viruses can be crystallized in the same way as proteins, and then showed that viruses are, in fact, proteins themselves.
What do the terms miscible and immiscible mean?
Miscible and immiscible are terms that refer to whether two liquids that come into contact will mix to form a single uniform solution. Two miscible liquids are able to mix together to form a uniform solution, while two immiscible liquids will separate into two layers. If the liquids are immiscible, the liquid with the lower density will stay on top and the liquid with the higher density will be on the bottom.
How many more Earth-like planets might be out there?
It's hard to say, but considering the tremendous number of stars out there, see below, it's possible that there are a lot.
Why did Melvin Calvin, United States, win the Nobel Prize for Chemistry?
For his research on the carbon dioxide assimilation in plants. Calvin received the prize for his work on what is today known as the Calvin cycle, which is the process by which green plants affect the fixation of carbon dioxide. Or in other words, the incorporation of carbon dioxide molecules from the atmosphere into other molecules. Calvin identified that there is a close connection between the metabolism of carbohydrates and photosynthesis. This process is quite complex, involving 10 different intermediates and 11 different enzymes to catalyze each step of the reaction.
What are some of the first organometallic complexes, and when were they discovered?
Among the earliest organometallic complexes was a compound known as cacodyl, or tetramethyl diarsine. This compound, with the chemical formula C4H12S2, shown below at left, was first discovered in 1760 and was known for its toxicity and terrible odor. The first platinum olefin complex, C2H4Cl3KPT, also known as Zeiss salt, see below, right, was discovered in 1829. This early organometallic complex was influential in establishing some of the essential underlying concepts that have since become crucial to organometallic chemistry.
What absorbs liquid in diapers?
The general term for the materials that absorb water in diapers is superabsorbent polymers. These same compounds are used to chill drinks and to make fire retardants and fake snow. Modern absorbing materials are typically sodium salts of polyacrylic acids, and these can become almost entirely water by weight and as much as 30 to 60% by volume.
What are auxetic materials?
Auxetic materials have a unique property when they are stretched. they actually become thicker in the directions perpendicular to the applied force. Think about this in comparison to anything else you stretch, it is really quite a strange phenomenon. This occurs as a result of hinged arrangements within the material that flex apart when a force is applied to stretch it.
Why did Hans Fischer, Germany, win the Nobel Prize for Chemistry?
For his researches into the constitution of hemin and chlorophyll, and especially for his synthesis of hemin. Fischer was interested in biologically relevant pigments, specifically those in human bodily fluids like blood and bile, and the green color of plants. He was the first to determine the correct structure for heme B and heme S, which are iron porphyrin molecules, a large ring with 4 nitrogen ligands coordinated to an iron center. These red-colored molecules help transport O2, and other important functions. He also determined the structure of chlorophyll A, which has a structure similar to that of a heme. But instead of an iron, a magnesium ion sits in the middle of the large porphyrin ring.
What is a chalcogen?
A group 16 element, oxygen, sulfur, selenium, tellurium, polonium, or livermorium.
How is nuclear chemistry different than other types of chemistry?
As the name implies, nuclear chemistry deals specifically with chemical events involving the nucleus itself. While most other areas of chemistry involve rearrangements of the electrons, nuclear chemistry is focused on radioactivity and the properties of nuclei, and it finds some of its most important applications in energy production, weapons, and medicine.
What is an adiabatic?
A process that does not absorb or release energy.
What is pyrophoric?
A substance that tends to ignite readily and spontaneously upon exposure to air.